The background to this project is that for hundreds of years people have known that venoms affect the cardiovascular system. The first ever high blood pressure molecule, captopril, came from a snake venom that caused transient low blood pressure in the victims. A lot of rattlesnakes, particularly from various regions of North America, on envenomation they cause significant local clotting of the blood, which causes significant downstream pathologies. Coupled with that, certain snakes also cause hemolysis, breaking open the blood cells, but also hemorrhage and various other bleeding disorders. And people have known these for hundreds of years. What isn't known is, does the venom have any effect on the actual hemoglobin? We know that hematophagic animals, such as the leeches and mosquitoes, they have hemoglobinase enzymes within their digestive system because they're actually feeding on blood. They need to break down the hemoglobin, remove the iron core to actually make use of the energy within the protein. But this is not a method of feeding for the snakes. But having said that, if the blood can be slowed down, then the snake can actually get an advantage to actually prey capture. So the two methods of prey capture with the snakes are clotting of the bloodstream. So when a rattlesnake bites a mouse, you'll typically see systemic clotting within a couple of minutes. And very clearly, once your bloodstream's clotted, the animal can no longer run away, and the snake is then free at its leisure to find the mouse and go and eat it. The elapids, the other group of snakes, the cobras and mambas, they act by neurotoxic paralysis, stopping the animal's breathing by paralysing the nicotinic acetylcholine receptors in the muscle nerve terminals. So again, respiratory paralysis coming in a couple of minutes from the dose in a rodent. In humans, this will typically take effect in about 15 minutes from a heavy envenomation. And once you can no longer raise your ribs and your diaphragm on your own, you stop running away very quickly. But once we were sort of looking at, okay, we don't know if it has any effect on hemoglobin. We obviously have a large library of venoms, great opportunity to have a look. So what we've done is we took our venoms, typicalised by this nice Western diamondback over here, and very simply paired them together with lysed hemoglobin. So we just took whole blood, mixed it at a 6:1 ratio with water, sterile water, and that lyses the erythrocytes. We then spin out the platelets, cell debris, and what you're left with, although quite crude, is a very high percentage hemoglobin solution that we can then work with. Initially mixing the venom with the hemoglobin, over the course of 5 or 10 minutes we didn't really see any activity, but then overnight we saw that several of the wells had actually changed colour, and the blood itself did not go off. It was purely a venom effect. We then talked to our friends at BMG LabTech to see if we could actually, if one of their instruments could actually look at this experiment for us. And the first tool we used was BMG Galaxy, and we picked two different wavelengths, 492 and 595 nanometers. These correspond with the absorbance levels of hemoglobin. And then we set about putting a plate. So our T-VDAcv, our targeted venom discovery array for cardiovascular disease, that's what the acronym stands for, which is mainly a plate based on snake venoms because we know they are the strong lead material in this area. And incubating them with the lysed blood, taking readings every 10 minutes overnight, so over a 16-hour period. And what we see is an increase in absorbance at the two wavelengths we first looked at. Not necessarily what we thought we were looking for at the beginning. We were expecting to see a decrease, a degradation of the hemoglobin, and what we actually saw is an increase in absorbance. So I'll take you through the plate. So 96 well plate, A1 in the top corner there. The top two rows are duplicates of the vipers from North America. We know these have very strong activity on the red blood cells themselves. But what we also see is we do see activity in a hemoglobin modifying process. And at that stage we were unsure of what was actually going on because we've got the opposite to what our expected results were. When we got into the cobras and mambas, the green and purple, there's only one venom that stands out above all the rest. And to find any in that group was quite a surprise. These animals are acting by neurotoxic paralysis. They have very minor hemodynamic effects on envenomation. Some of them do have tissue destructive effects and cytotoxicity. And actually, this is one of those that stands out. This is a black-necked spitting cobra from Africa, a Naja nigricollis. Our animals are 8 foot long, quite angry, stand up and fight for themselves. But very interesting, the neurotoxic paralysis, if you survive that, and it's quite easy to actually provide respiratory support for people to actually then, such that they can survive the neurotoxic paralysis, you actually see a lot of tissue destruction, and this is through three-finger toxins actually destroying cells in the basement membrane. So we were quite surprised and quite excited to see an elapid venom having a direct effect on the blood, although at that stage we were unaware of what it was. The bottom line, in the darker red and blue, these are our old world vipers, things like your adders and their close relations in Northern Europe, but also puff adders and gaboon vipers of North America that deliver huge doses of hemoactive venoms. So our focus was on the cobra venom. We got a large source of this, and it is completely novel because it's an activity we didn't expect in elapid venoms. We nicely saw an absorbance at 492. It was just measured over time, taken out to 15 hours, and what we see is that a dose-responsive reduction in activity going on dilution of the venom. So the venom comes out of the snake typically about 300 mg per mil, very high concentration protein solution, and as a result we got very high dilution activities and still got significant effect. But we were still unsure of what this effect was. It was opposite to what we were expecting. We were looking for a degradation of the hemoglobin, and what we're actually seeing is an apparent increase in the absorbance. There was no precipitation events, it was a direct increase in hemoglobin absorbance. We then moved on to use the Ferrostar galaxy, and this gave us a significant advantage because we can actually scan the wavelength from 400 to 700 nanometers. It gives a full profile of what's happening. So our 495 and 595 activities are within that region, and we can actually get a full profile. So this is just our lyse blood, our crude hemoglobin solution, incubated with PBS overnight, and with the lines overlaid on top of each other. So there's no error bars here, you're seeing the full data set. And what we see is that there's a little bit of degradation over time, but very little measurable effect. When we then dosed with the venom and done the same thing in a side-by-side well, what we see is a very dramatic effect on the spectrogram. In these areas here, where our initial wavelengths were, we see a significant time-responsive and indeed dose-responsive increase in the absorbance at the nearly 500s and the near 600 areas where we were looking at before. And quite diagnostic is the shoulder coming in at 625 in the later period. The two middle peaks are connected with the oxidation state of the hemoglobin and the amount of oxygen it's carrying, and you see a decrease in this area. So the wavelength scan opened up a lot of what was going on. And what happens if you actually look at the Zylstra and Bursa data, the reds don't come out quite so well on the presentation. But you can see here the typical twins peaks of a standard oxyhemoglobin state. When you remove the oxygen, the blue peak, you see a dipping in these areas and those twin peaks coming to a single peak once you remove the oxygen from the bound hemoglobin. And then what's really important to us is this change here, which is a change to met hemoglobin. So what's happening here is the iron 2 from the hemoglobin core is being oxidized to iron 3. This happens naturally in the body, but only to about 10%, and is cleared up by the liver quite quickly. And this, obviously, as you see, this profile matches perfectly with the profile we see later on. And in the body, we've got about sort of 15% met hemoglobin. Doesn't say any significant changes. Once you start up to get 50%, you see the profile starting to form. So what we're realizing here is that, along with the neurotoxic paralysis, the snakes have evolved a longer-term mechanism that stops hemoglobin binding oxygen, and therefore an additive effect in the process. And it's interesting for us is that we've not found any recollection of anything in the literature of a venom or a toxic secretion from an animal actually changing the iron oxidation state in haemoglobin. Things, sort of various weed killers like paraquats, lidocaine, and several other compounds will cause this effect in patients. It's diagnosis of seeing blood that looks brown, and if you see on our poster, you'll actually see the plate colour. The blood does turn from a nice bright red into a browny colour. And that is expected because once you oxidise iron from iron 2 to iron 3, you start to see that rust colour, and that's exactly what you're seeing in the blood. So by using the Ferrostar, we're actually unpicked some novel biology in some of our venoms, which is quite exciting that we'll be taking further to find out more about is this an anecdotal effect, or is there an evolutionary pressure for modifying haemoglobin for difficult to capture prey, for instance. And so yeah, and come see me at poster 78 with the full detail. And we've got copies on our stand A5 as well. Okay, thank you very much.